How did electroencephalography, EEG, first develop? Imagine being able to listen to the brain's electrical whispers. That's what early scientists wanted to do when they started exploring brain activity. The story begins with curiosity about how living tissues produce electrical signals. In the late 1700s, scientists like Luigi Galvani discovered that electricity could make frog muscles twitch. This showed that living things generate electrical signals, laying the groundwork for bioelectricity. But it would take many years before these ideas were applied to the brain itself. In the late 1800s, researchers such as Richard Cadden used devices called galvanometers. These devices could detect tiny electrical currents. Cadden took these instruments and placed them on the exposed brains of animals like rabbits and monkeys. He observed that brain activity changed when the animals saw or heard things. This was the first clear evidence that the brain's electrical signals could be measured and might relate to mental states or sensory experiences. The real breakthrough came in the 1920s with a German psychiatrist named Hans Berger. He wondered if he could record brain activity from a person's scalp. Using a very sensitive galvanometer, he succeeded in doing just that in 1924. This was the first human electroencephalogram. Berger noticed different patterns in these brain signals. One pattern, called the alpha wave, appeared when a person was relaxed with their eyes closed. Faster waves, called beta waves, showed up when the person was actively thinking. Berger also saw that alpha waves disappeared when the eyes opened, linking brain activity to behavior. At first, many scientists doubted Berger's results. It took several years for others to confirm his findings. By the mid-1930s, scientists in England and the United States repeated his experiments successfully. EEG started moving from a research tool to a medical instrument. The first clinical EEG lab opened at Massachusetts General Hospital in 1937. Doctors began using EEG to identify abnormal brain activity, especially in epilepsy. They saw rhythmic spike wave patterns in seizures, which helped them connect symptoms to specific brain signals for the first time. Technological improvements played a big role in EEG's growth. Early devices used mechanical galvanometers, which were slow and not very precise. In the 1930s, electronic amplifiers and oscilloscopes made recordings clearer and easier to analyze. These advances helped EEG become a standard tool in hospitals and laboratories. Today, EEG remains a core part of neurotechnology. It is used to diagnose neurological conditions like epilepsy, to study brain functions, and even in brain-computer interfaces. Small electrodes placed on the scalp detect electrical signals from the brain. These signals are amplified and displayed as waves, giving real-time information about brain activity. While imaging tools like magnetic resonance imaging and computed tomography scans show the brain's structure, EEG shows how the brain works in the moment. From animal experiments to modern neurotech, EEG's history shows how curiosity and innovation can reveal the brain's electrical language. It's a story of discovery that continues to shape how we understand and interact with the human mind today.